Experience 5 goes over the identification of microorganisms where we're looking at gram-positive cocci and bacilli. So we're going to start looking at some of the differences we get in here. With gram-positive bacillus, what we're looking for really is the presence of spores. Are there endospores present versus are there no endospores? Because this really gives us an ability to break things down. Endospores look like these. The top two are gram stains where you can actually see the endospores on the inside of the cells, and you see them not because you're staining the endospore, but because the endospore doesn't take up stain very easily, and therefore we see that. Endospores are very important because they allow for the organism to survive long periods of time. Bacillus and Clostridium tend to have spores present. The top two are Bacillus. The spores are about the same diameter as the cell. The bottom one is Clostridium because the endospores are a much greater diameter than the cell. Corynebacterium does not have spores. So we break these apart based on whether or not they've got spores. If we look at gram-positive cocci, we start off saying there are no spores. We run a catalase test on them to separate out the two major groups. It can either be catalase positive, in which case we get foaming. Catalase positive means we are going to run it on mannitol salt agar because we've got a staphylococcus. Or it could be catalase negative, which means we get no appreciable foaming out of it. And the negative we run on blood agar because that's going to be a streptococcus. If it's catalase positive, we're looking at staphylococci. With staphylococci, we're going to do a mannitol fermentation test on mannitol salt agar, which also looks at tolerance to the high salt concentration inside of the mannitol salt agar. We're basically looking for growth or no growth, and we're looking at the ability to ferment mannitol. Here is a plate of mannitol salt agar. On the left-hand side, we can see how the mannitol has changed from a nice, pretty pink color to this very yellow color, which is a positive result. Therefore, the material on the right-hand side is going to be positive for mannitol salt agar, and that is Staph aureus. On the left-hand side, you're going to have the pink color that it originally started with, and that's Staph epidermidis. When we look at catalase negative, we're going streptococcus. Streptococcus means we're going to run it on blood agar, and we look at hemolysis on blood agar. We have beta or complete, what we call total hemolysis, and that would look like this. When you look at these two plates, look at the edge of the colony and the edge of the zone of clearing, and you can see how they are separated out. That distance between the two, we know that this is beta or complete hemolysis. We have what we call gamma, which is no hemolysis, which means that when you look at the growth, you do not see any change in the agar itself. And we have alpha, which is a partial hemolysis. And with the alpha, hemolysis that you get is minimal. It's right on the edge of the colony, and we start to get kind of a greenish color. It really doesn't destroy the erythrocytes. It just causes some of them to break apart, so to speak. So we've got the three different types of hemolysis. Very important to understand that. CAMP test. CAMP factor, it differentiates strep group B from strep group A. Most S. agalactias strains produce a diffusible extracellular compound that will, in conjunction with the specific beta-hemolysin of Staph aureus, cause complete lysis of sheep red blood cells in an agar medium. Let's translate that. What we've got here is a streak of bacteria down the center. That is a Staph aureus. And away from the Staph aureus, it produces a beta-hemolysin that basically diffuses out into the agar. Now, on the left-hand side of this, about 3/4 of the way up, you can see that there's another streak going out to the left. And about 1/4 of the way up on the right-hand side, you can see that there's another streak coming out from the right. Where these two parts come together, we see a change in the coloration, which means we've got beta hemolysis. It gives us an arrowhead type pattern, and therefore this is really what we would call a positive CAMP test. If we had a negative CAMP test, we would find the material coming right up to the staphylococcus in the middle, and you'd get no color change. So this is a positive CAMP test. We also have positive test for NaCl. It says we run it on a 6.5% NaCl broth. We look for turbidity within 72 hours, and you can see that the right-hand tube has turbidity. The left-hand tube is clear. We run bioesculin. A positive reaction is a dark brown to black color, which is hydrolysis of the esculin. In 18 to 24 hours, you can see the left-hand side shows you what the tube looks like uninoculated, and the bioesculin positive is going to give you this nice dark brown to black color. And we also have antibiotics, and with the antibiotics, we record a positive if we get a clear zone, which shows sensitivity of the bacterium to the antibiotic. We're looking at a place where we get no growth. The bottom two discs here have inhibited growth of this particular bacterium. Therefore, that would be a positive result. We take all of those and we run them on this particular chart, which looks at Lancefield groupings for Streptococcal ID. It takes into account hemolysis on blood agar, CAMP test, bioesculin, NaCl tolerance, sensitivity to the antibiotics SXT, Optocin, or bacitracin. And based on this, we can come out and we can determine which group each organism would therefore fit. And this becomes very interesting because it's the way we differentiate things like pyogenes, which causes strep throat, from agalactia, which is normally not found in the strep throat area.